For this lab, we're going to be covering 3 body systems in one single lab. And the reason we're covering all 3 together is because they're tied together by what they do, and that they all help maintain what we call homeostasis, or the ability for your body to maintain a relatively constant internal environment. So let's look at these 3 body systems and how they maintain homeostasis. Homeostasis, like I just mentioned, is this large-scale process, an across-the-body process where you have to maintain an internal balance. The conditions inside your body have to stay relatively constant. Now, that doesn't mean that everything has to stay perfectly still. It doesn't mean your temperature has to be 98.6, and if it goes to 98.7, you die. However, there are certain parameters we have to stay within. If your temperature rises to 101, even though that's just a couple degrees higher, that can cause major problems within your body. Now, we're not going to get into all of the ways that our body maintains homeostasis. That's something that we'll check in with as we go through the semester, mainly in lecture. However, we do want to introduce this idea and know who's kind of in charge and how we can control for these parameters. So there's 2 main body systems that are in control of homeostasis: the nervous system and the endocrine system. However, this lab is not about those. We'll talk about those in different labs, but we just wanted to introduce that idea. The endocrine and nervous systems are going to exert control over all the other body systems and influence them to do activities that help us maintain homeostasis, which we'll see in the 3 body systems we're going to be looking at. All of these systems use processes of positive and negative control to maintain body conditions. Positive control is where we're kind of going in a certain direction, we're going to continue in that direction. And in some cases, this is really helpful. So for example, when a woman is in childbirth, she will release oxytocin to promote uterine contractions, and those contractions will also promote the release of oxytocin, which then feeds into itself, so that as we release more oxytocin, we have more contractions, and the more contractions we have, the more oxytocin. It's like a snowball effect. Most things, though, are controlled by negative control. Negative control is kind of like a teeter-totter or a stocking the shelves kind of thing. The teeter-totter example is where we're trying to maintain balance in the middle, and if we tilt too far in either direction, we have some sort of mechanism to bring us back into that middle balance. If we go too far the other way, we have something that brings us back to that middle again. So we're always trying to maintain that teeter-totter balanced in the middle so that neither end goes up or down. And if it does start to go up or down, we have a way to bring us back to that middle ground. The stocking the shelves method is something we see we'll go into with the endocrine system, where a certain product is being made, and after we've reached a limit, we've reached a certain amount of that product being made, that product will then influence the beginning of that cycle to stop production. Kind of like if you worked at a grocery store and your boss told you to put cans of corn on the shelf. Once you filled the shelf to capacity, you would take any extras back to the back stock room and let your boss know that it is fully stocked. You wouldn't keep putting more cans of corn on other shelves or on the floor. The 3 body systems we're going to look at is the urinary system, the digestive system, and the respiratory system. These are in no particular order, and it doesn't necessarily follow your lab's order, but all 3 of these are going to be important for maintaining homeostasis in different ways, and they're in no order of importance. So we're going to start with the urinary system today in this video, where we're going to look at the various organs and hyper-focus on one of these organs. So let's look at the organs of the urinary system. We have the kidneys, of which you have 2, and these are going to filter out waste products from the bloodstream and create urine with those. The ureters connect the kidneys to the bladder and transport that urine down. And again, you have 2 of them, 1 ureter for each kidney. We then have 1 single bladder, which will take that urine and store it until micturition, also known as urination. And then lastly, we have the urethra, which connects our bladder to the outside of the body so that we can urinate. When it comes to maintaining homeostasis with the urinary system, we're mainly focused on the kidneys and their ability to take waste products and contaminants from the body and remove them. So they're going to filter the bloodstream and produce urine with those waste products. Here we see a cross-section, or I should say more of a longitudinal section of a kidney. You're not going to have to know all of these terms, so if you're looking at this kind of worried, don't worry. We're going to talk ourselves through just a couple things in this kidney. In lecture, you may go a little bit deeper, so just be aware of that. So let's talk about some of the major structures of this kidney. So here we have the kidney. Again, it's like a bean-shaped structure. Which came first, the kidney bean or the kidney? I do not know. And we have a few layers to this kidney. On the very, very outside, we have the renal capsule, which is kind of encapsulating the entire thing. In this pinkish color we have here, we have the layer called the renal cortex. In this more middle layer that has these kind of rounded out structures here, we call that the renal medulla. And one of these pieces here we call a renal pyramid. So we have the capsule, that's the most outer portion, the cortex here, the medulla closer to the middle, and one of these pieces being called the renal pyramid. The renal medulla and pyramids are going to drain here into the renal pelvis, which is this area right here, which then pretty much just dives right into the ureter. It's pretty continuous with that renal pelvis. We also have some blood vessels that help feed this kidney. We have the renal artery here in red and the renal vein here in blue. But we haven't quite talked about what actually produces the urine yet. How do we make urine in a kidney? Urine production is controlled by 3 steps: glomerular filtration, tubular reabsorption, and tubular secretion. Urine production is happening inside of a structure we call the nephron. The nephron is a microscopic unit found within the kidney, of which each kidney has about 1 million. These are the workhorses of the kidneys, the ones doing the filtering, doing the urine production process. So let's start with step 1, glomerular filtration. A blood vessel is going to enter into the glomerulus, a structure found within that nephron, which we'll look at it in just a second. And small particles are able to filter out of that blood vessel and into a capsule surrounding the glomerulus. When things filter out, we can only filter out things that are small. Larger things like blood cells and proteins are stuck inside the bloodstream, but smaller things like salts and water and urea can filter out very easily. As we move through the nephron, we get to step 2, tubular reabsorption. The filtrate, the things that filtered out in the first step, are going to enter into a section called the proximal convoluted tubule. Things like glucose, amino acids, salt, and water that filtered out originally are still useful, and so we reabsorb those back into the bloodstream. We don't want to get rid of things that still have a purpose or still can be used for something in our body. Glucose, amino acids, salt, and water are all still very useful things, whereas urea and uric acid are not. So those 2 things are going to stay inside the nephron. Lastly, we get to our 3rd step, tubular secretion. Things like hydrogen ions, as well as metabolites from drugs like penicillin, are going to be secreted into the nephron, joining up with the urea, ammonia, some water, etc. as urine. This helps us maintain pH balance of the blood. Hydrogen ions are acidic, and so by pulling those acidic components out of the blood, we can keep our blood's pH at about a 7.4. So here's our nephron here. Let's look at some of the components. So here is our blood vessel that is bringing in blood to be filtered out. We call this the afferent arteriole. Think afferent arrives, that might help you. Here is the glomerulus, the kind of blood vessel knot we see inside, and it's surrounded by this glomerular capsule, also known as a Bowman's capsule. So when we're in here, glomerular filtration takes place. We can filter out those teeny tiny things like glucose and salts, waste products like urea, as well as things like water. So those all filter out and enter into the capsule. Any blood that is still within the bloodstream, anything left inside this glomerulus, is going to leave the glomerulus out of the capsule through the efferent arteriole. Think efferent is exit. That might help you remember those. All of the filtrate, the things that filtered out and entered into the capsule, then enter into the proximal convoluted tubule. You can see why it's called that. Proximal because we're in close proximity to the capsule, and convoluted because we are kind of going in all these different directions, right? We're twisting and turning around. From there, we dive deep down into the loop of nephron, also known as the loop of Henle, and then back up. While we go through the proximal convoluted tubule, the loop of nephron, and then the distal convoluted tubule, which is right here, right, because it's in the distance of the capsule, we're going to reabsorb those things that are still useful. And we're reabsorbing them back into the bloodstream. So you can see how we have these capillaries that are kind of twisting around the tubules. They're twisting around the nephron here. They're in close contact or in close enough contact that they can reabsorb things like water and salts. And then lastly, we get to what we call the collecting duct, which will be connected to many nephrons. So like here, 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 and here. That would connect to other nephrons nearby. And by the time we get to this point, we are pretty much urine. We can do a little bit of recollecting water here, and we can do some last minute secreting of things like medication byproducts and hydrogen ions as we go through this section here. So we can do a last minute secretion of other components that we don't want to keep in the body here. These all will collect and become part of that renal pelvis, and then finally become part of that ureter. Again, this is the microscopic scale though. So on that kidney we saw earlier, you have about 1 million of these in each. Since what constitutes waste is pretty particular, and there's certain things we don't want to see in urine, a urinalysis is a tool that we can use to check on the kidney functions. The things we mentioned earlier, things like blood cells and proteins and glucose, are things that don't normally end up in the end urine product. And so if we see those in the urine, that can tell us what might be going on, specifically what might be going wrong with the kidneys. So glucose, proteins, blood, if that ends up in the urine, that can tell us something is wrong. And there's various things that can go wrong with the kidneys, and your lab manual goes into that depending on what material you find in the urine. A urinalysis is kind of like a pool test kit. Basically, you collect a urine sample in a little cup. You have a dipstick that has various things that it's looking for. You dip the dipstick into the urine, and then you look and see for certain color changes. It can tell you if something's within a normal range or if something is an abnormal range. Let's move on to the second of our 3 homeostasis or maintenance systems, the respiratory system. This is another way that we can get rid of some byproducts we don't want, namely carbon dioxide. The lungs are responsible for this gaseous exchange of nutrients and waste, meaning we're bringing in fresh oxygen molecules and getting rid of carbon dioxide. This is happening at a microscopic level, where we have alveoli, small little pockets of simple squamous cells that can expand and contract due to pressure we see in the chest cavity. Here we see the lower part of the respiratory system. We have the trachea that leads down and splits into 2 bronchi, one bronchus going one way and the other going the other way. Each bronchus connects to a lung. So we have a lung here and a lung over here. Those bronchi are going to branch out into bronchioles and get smaller and smaller and smaller into smaller branches. And at the ends of these branches, we find those alveoli, those little air sacs where we can perform that gas exchange. Each lung will also have blood vessels leading into them, the pulmonary arteries in blue and pulmonary veins in red, since the right side of the heart will be sending blood through these lungs to get oxygenated and to dispose of that carbon dioxide we don't want. And if we look at the lungs, something to kind of quickly point out, we see that are divided into these little sections we call lobes. On the right side, again, remember from the last lab that right and left are flipped when we're looking at an organ head on. On the right side, we have 1, 2, 3 lobes, and on the left, we only have 2. Notice that the heart kind of moves in and takes some of the space away from the left lung. So the left lung is on the smaller side compared to the right, and it's that way because we have to make space for this heart. Something to pay attention to. Let's get into breathing. Now, you may be familiar with terms like breathing in and inhalation and exhalation or breathing out, but we have some other terms we prefer to use in biology: inspiration and expiration. Inspiration is the breathing in, and expiration is the breathing out. When we bring air into our lungs, we're using negative pressure. Basically, we force the chest to expand, which drops the pressure inside of the chest, and air gets pulled inside. When we shrink that space by having the diaphragm move up and in and the ribs move down and in, the space, then the pressure becomes higher, which pushes and forces the air out. So when we inhale, and it might be useful to kind of do this with me, when we inhale, your diaphragm is moving down and flattening out, your ribs move up and out. Your entire chest cavity gets larger. Because it's gotten larger, the pressure inside of your chest cavity goes down, and that pulls air in to equalize the pressure on the inside and outside of your chest cavity. When we want to exhale, notice your diaphragm moves back up and in, and the ribs also move down and in, shrinking the entire chest cavity. When that happens, we're increasing the pressure we have inside the chest cavity. And since the pressure is lower on the outside of your body, air gets pushed back out. This is how we breathe and make sure that air is constantly moving in and out. These are the actions we perform to allow air to move through the respiratory system. Right, and so if we look at the entire respiratory system together, right, we breathe in, you can't see the ribs here, but they would move up and out. This diaphragm here would move down, and that would increase the volume we have inside this chest cavity. Whereas when we are breathing out, the diaphragm moves back up into this kind of C shape. The ribs would move down, back and in, shrinking the volume we have here, putting higher pressure inside this cavity, which would force air up and out. Now let's move on to our last maintenance system, the digestive system. The digestive system's main focus is to bring in important nutrients that we need, building blocks for all of the things that we have within our body. In order to do that, we have two sections: the gastrointestinal tract, or the GI tract itself, where food will actually pass through, and the accessory organs, important organs for digestive processes that don't actually physically come into contact with the food we eat. In the GI tract, we have the mouth, the esophagus, the stomach, the small intestine, the large intestine, and the rectum and anus. For the accessory organs, we have the liver, the gallbladder, and the pancreas. Overall, our goal as the digestive system is to bring in food, break it down to its small components, absorb those components, and eliminate anything left over. So let's get into the GI tract first. We start with the mouth, which will help break down food via mechanical and chemical digestion, meaning we physically break apart the food by doing things like chewing, as well as chemically start to break down the food using enzymes found in your saliva. We then need to transport that food we've chewed up down towards the stomach, and we do that using the esophagus. The esophagus will move food from the mouth down to the stomach through a process called peristalsis, meaning rhythmic muscle contractions that will squeeze that food downward. Once we reach the stomach, we can further break down food, and we can do that using mechanical means where we're physically using muscle to mash and stir things around, as well as enzymes like pepsin that break down proteins. The stomach will also store food and slowly release food to the small intestines. This can take about 2 to 6 hours for a single meal, where the stomach will hold on to it and slowly release little bits of it so that your small intestine isn't overwhelmed with a large amount of food. Once we receive that food in the small intestines, we're going to further break down that food using chemical means, where we use enzymes to break down the food into its smallest units. Once we're in those small units, we can absorb those nutrients directly into the bloodstream. After that, we send that food, or what's left I should say, to the large intestines. The large intestines are going to absorb water as well as absorb some small vitamins here and there and form the feces, the waste products we don't want to keep anymore. Lastly, we get to the rectum and anus, where we can store those feces away until we can eliminate them properly. And so we look at the image over here, we see this pathway of food, right? We bring food into our mouth, we chew it up, we swallow down through the pharynx and esophagus, down to the stomach, to the small intestines, the large intestines, and then finally the rectum and anus. The accessory organs are still very important for digestion. However, they don't have physical contact with the food we eat. And we have 3 accessory organs: the liver, the gallbladder, and the pancreas. We're going to focus on the functions that these 3 perform in relation to digestion, although it's important to note things like the liver do have other functions that are unrelated to digestion. We're just not going to get into those right now. So when it comes to digestion, the liver will help by producing bile, which will help break down fats, as well as store nutrients like glucose and certain vitamins. When the liver produces bile, it sends it down to the gallbladder, and the gallbladder will hold on and store that bile, as well as control its release. So we look at the image, we see the liver here. It's one of the largest organs you have in your body. And the gallbladder, which is usually found kind of hidden underneath this side of the liver. They're really kind of tied together. They're very close to each other. Our last accessory organ is the pancreas. The pancreas produces digestive enzymes that it can send to the small intestines to break down food into its smallest units. It also will neutralize the stomach acid we find in the stomach, so that once that food that we've eaten goes from the stomach to small intestines, we neutralize that acid so that we're more neutral in the small intestines. Otherwise, it would burn. As the pancreas, we also regulate blood glucose levels. I'm not going to get into that in detail here, since we'll talk more about the pancreas in the endocrine system. But this is how we can relate all of these organs to the digestive process.